In this topic, we're going to discuss the membrane structure. So, by the end of this topic, you'll be able to describe and explain the fluid mosaic model and outline the roles of cell surface proteins. Now, cell membranes separate living cells from non-living surroundings, and they are a thin barrier about 7 nanometers thick. So they control what goes in and out of the cell. We say that they are selectively permeable. This means that they allow some substances to cross more easily than others. For example, hydrophobic substances can cross more easily than hydrophilic substances. They are made of phospholipids, proteins, and other macromolecules. Let's have a look at phospholipids. A phospholipid is made up of 2 parts. You've got your fatty acid tail, which are hydrophobic. So they don't like water very much, and they repel water. Phosphate group head, which is hydrophilic, and this likes water, so it's attracted to water. They are arranged as a bilayer, so this acts as a barrier to water-soluble substances. For example, bilayer, you can see one phospholipid at the top, another phospholipid at the bottom, with the tails facing each other. So this is an example of a structure-function example. Why is it arranged like that? So just another example of how the phospholipid bilayer is formed. You can see the hydrophilic heads are pointing outwards and the hydrophobic tails are pointing towards each other, away from the aqueous solution. So the polar hydrophilic heads are water-soluble and the hydrophobic heads are water-insoluble. So here you can see how the phosphate heads are pointing towards the aqueous solution and the tails are pointing away. So the tails are hydrophobic, the heads are hydrophilic. If you were to submerge this into water, you would see a micelle forming. So this is where the phospholipids have got their heads pointing outwards and the tails pointing inwards. Now in 1925, Orta and Grendel proposed that the unit membrane is formed from a phospholipid bilayer. What does this mean? Well, you've got one layer of phospholipids with the hydrophilic heads pointing inwards towards the cytoplasm, and the other layer of phospholipids with the hydrophilic heads pointing outwards towards the water which surrounds the cells. The hydrophobic tails of both layers are pointing towards the center of the membrane. Now, the more saturated these tails are, the more fluid the membrane is, which we'll look at in a little bit. So if you were to put phospholipids in water, they would form one of 2 structures. They'd either form a micelle, which you can see here, the phosphate heads are pointing towards the water and it's formed a circular structure, or bilayer. So in each structure, the hydrophilic heads face the water and the hydrophobic tails point inwards, away from the water. This behavior is key to the role that phospholipids play in membranes. 1972, Singer and Nicholson proposed that the membrane proteins are inserted in the phospholipid bilayer. So, how do we get the name fluid mosaic model? So it's like a fluid, it's like a mosaic, so it must be a fluid mosaic model. Now the membrane is a collage of proteins and other molecules embedded in the fluid matrix of the lipid bilayer. You've got your cytoplasm, extracellular fluid, and you've got your lipid bilayer sitting between. So you've got the filaments of the cytoskeleton, phospholipids, cholesterol, transmembrane proteins which span the whole bilayer, peripheral proteins which sit on the outside, glycoprotein is attached to a carbohydrate group, and then a glycolipid is attached, has got a carbohydrate group attached to the lipid. If you were to draw it in simple terms, you can see the different parts. Let's start with the peripheral protein. Notice how it's on the outside. You've also got enzyme or signaling proteins. Integral protein spans the whole membrane, as does a hydrophilic channel within a carrier protein. You've got a glycoprotein which is attached to a carbohydrate chain, and then some proteins act as receptors for recognition by hormones. So the fluid mosaic model of the plasma membrane, the proteins can move freely within the lipid bilayer. The ease with which they do this is dependent on the number of phospholipids with unsaturated fatty acids in the phospholipids. So the more unsaturated they are, the more fluid it is. Let's have a look at cholesterol. Now, cholesterol molecules occur throughout the cell surface membrane of animal cells. They are very important and they help with mechanical stability. So here you can see a cholesterol molecule. It's got the molecular formula C27H46O. And this prevents ions and polar molecules from passing through the membrane. So cholesterol is very important in controlling membrane fluidity. The more cholesterol you have, the less fluid and the less permeable the membrane. So it's important in keeping the membrane stable at normal body temperature. Without it, cells would burst open. How it does this is it helps to pull together the fatty acid tails of the phospholipid molecules, limiting their movement but adding mechanical stability. Let's have a look at the different membrane fat composition. So the fat composition affects flexibility, and we know that the membrane must be fluid and flexible. So it's about as fluid as thick salad oil. So how is this varied? Well, this depends on the percentage of unsaturated fatty acids and phospholipids. These keep the membrane less viscous. For example, in cold-adapted organisms like winter wheat, the percentage increases in autumn. You've also got cholesterol in the membrane. So here you can see unsaturated hydrocarbon tails make the membrane more fluid, and viscous tails make it more rigid. Okay, let's have a look at the membrane proteins. So the proteins determine the membrane-specific functions. You've got cell membrane and organelle membranes. Each have unique collections of proteins. These include peripheral proteins. Peripheral proteins are loosely bound to the surface of the membrane, and they act as cell surface identity markers, for example, antigens. Then you've got integral intrinsic proteins, so they penetrate the lipid bilayer, and they usually span the whole bilayer. They act as transmembrane proteins or transport proteins. For example, they might have channels or they act as enzymes. So you've got, when you have a protein sitting inside the membrane, the non-polar amino acids are going to be facing towards the membrane. So these are hydrophobic. They anchor the protein within the membrane, and they're on the outer surface of the membrane. Then you've got your polar amino acids, which are hydrophilic, and these extend in the center of the protein and into the cytosol. So proteins typically make up about 45% of the cell membrane, and they do vary. So you've got your integral. Remember, the integral goes throughout the whole protein, and then the peripheral protein. Peripheral proteins are confined to the inner or outer surface of the membrane. And then you do have some glycoproteins with the carbohydrate chain attached. Let's have a look at integral proteins. Many integral proteins are carrier molecules or channels, and they help to transport substances such as ions, sugars, amino acids that can't diffuse across the membrane but are still vital to the cell's functioning. Other integral proteins are receptors for hormones and neurotransmitters, or enzymes for catalyzing reactions. So your integral proteins can act as carrier molecules or channels, and they also act as receptors for hormones, or they can act as enzymes. Peripheral proteins, they sit on your outside. They may be free on the membrane surface or bound to an integral protein. Peripheral proteins on the extracellular side of the membrane act as receptors for hormones or neurotransmitters, or they're involved in cell recognition. So we say that many of them are glycoproteins with that carbohydrate group attached. Peripheral proteins on the cytosolic side of the membrane are involved in cell signaling and chemical reactions. They can dissociate from the membrane and move into the cytoplasm. So you've got lots of different functions of membrane proteins: transporter, enzyme activity, cell surface receptor, cell surface identity marker, cell adhesion, and attachment to the cytoskeleton. We'll just quickly look at membrane carbohydrates. So these play a role in cell-to-cell recognition. So the ability of a cell to distinguish one cell from another. They can act as antigens, and they are important in organ and tissue development. So they do form the basis for rejection of foreign cells by the immune system. They act as receptor molecules, for example, for hormones and neurotransmitters, and antigens, allowing cells to recognize each other. So why do we call the phospholipid bilayer a fluid mosaic model? Well, it's fluid because it's made up of phospholipid molecules that can move. This means that it's flexible and it's constantly changing shape. It's mosaic because the proteins that are embedded in it vary in shape, size, and pattern in the same way as the stones or tiles vary in a mosaic. And then lastly, what is the use of the membrane? The membrane contains cell or organelle contents. It helps to compartmentalize organelles in the reaction, compartmentalize chemicals, for example, enzymes and toxins, prevent loss or entry of chemicals, and allows transport of certain substances in and out. It is also the site of chemical activity. For example, when we discuss respiration, the cristae on the mitochondria are the site of respiration. So, in summary, the cell membrane is described as a fluid mosaic model, and you've got different components. These are phospholipids, cholesterol, proteins, glycolipids, and glycoproteins. And that concludes our lesson. The end.